All right, well, thank you everybody for coming out on what I guess is a chaotic traffic night. That never happens here, right? I am so humbled to be here. I mean, this is an amazing, amazing audience. And I hope in sharing with you the work that my fantastic fellows and postdocs and graduate students have been doing over the last 4 years, I can get you excited about genetics and genomics, and in particular about the dogs. So people often ask me, why dogs? Why would you do genetics of dogs? And, you know, there are very practical answers. There are very large numbers. You know, about 90 million dogs are owned in the United States, and about 1 in 3 households own a dog. Dogs live in our environment, they eat our food, they get exposed to the same junk that we do, and that offers lots of opportunities for doing medical genetics. For me, the most important thing is their extensive morphologic and behavioral diversity. And I am so excited about understanding the differences between breeds in terms of all of those things. And we'll talk about a lot of that today. But I'm also at the National Institutes of Health, and we are very committed to human health as well as canine health. Dogs get the same diseases as humans. They receive extensive preventative and diagnostic health care. In 2022, I was surprised by this, but Americans spent $137 billion on their pets. And about half of that went to healthcare, to medicines, to vet visits, to vaccines, to diagnostics, to pet insurance, all those sorts of things. And finally, there's a very practical issue of sampling. You know, when we do human genetics, which my lab has done for many years, breast and prostate cancer, we're lucky if we can get a couple generations in a pedigree. In dogs, it's really not a problem to put together 5. And that offers us tremendous statistical power and opportunities that we don't have looking at the exact same traits in human families. So when it comes to projects, I'm a little bit like a kid in a candy shop. I am in love with everything that my lab is doing, and I always want to do something new and take on something new. So we do have a lot of projects, and each one is run by some incredibly talented trainee. I'll talk today about the work that we're doing in understanding how breeds are organized in relation one to another. I'll talk about morphology and body size, sort of obvious traits in dogs, but where those alleles came from 50,000 years ago, which surprised us. I'll talk about dog lineages, you know, what are the most diversified breeds and what are the things that distinguish them. And one good example will be herding, and we'll be talking a little bit about herding as well. I'll talk about a genotype-first approach, so getting a variant and then trying to work backwards and figure out a trait that it's assigned to. And I'll talk about the genetics of cancer susceptibility. This is actually a Bernese Mountain Dog. And I don't know if any of you know this breed, but about 25% of them will go on to get histiocytic sarcoma, and all who get it will die of the disease. So this is an important problem to solve in canine health, but it's also a tremendous system for understanding this and similar diseases in human health. And the red here highlights one of the loci that my lab described that contributes to the risk for this disorder. So people always ask me, sort of, where are you from? How did you get into this? And so I think you said all of it. I graduated from the University of Washington. I did my PhD at the Oregon Medical School. I was a Damon Runyon Fellow at Harvard for 3 years. And as both a graduate student and a postdoc, I worked on DNA structure and topoisomerases. I think I published the least cited paper ever on the dimensions of the minor groove of SV40 during replication. But I also published the most cited paper ever on how to get spilled DNA out of laboratory bench paper. So those are my claims to fame. I went to Berkeley, which is where I started the Dog Genome Project in Jasper Ryan's lab. And then I was tenured and tenure track faculty at the Hutchinson Cancer Center in the University of Washington. And as Luke said, I moved to NIH in 2004. But for the trainees in the lab, in the audience, I do want to make it clear that if you know where you want to be and you're looking at where you are today and they don't match up well, you can still get there. It's just, it's often a very unexpected road that you have to take to go from position A to position B. And mine was very unexpected. When I graduated from high school, I worked as a janitor, and I was a really outstanding janitor, I will point out. I picked fruit for 2 years in the Yakima Valley. I got a job as a dishwasher, from which I was almost immediately fired because it turns out glasses don't go down the garbage disposal. Well, that was a problem. But then I got this great job as an autopsy assistant, and that was really my first introduction to medicine, and really made me think about, oh, well, there's a lot of unanswered questions here that are exciting. So I got a low-level job at the University of Washington, but that advanced to be a low-level technician, and that was it. That was when I knew I was going to be in the lab, and I just had to figure out how to get from here to there. But even getting to that point had so many challenges and so many weird steps for me. And I want to encourage the trainees who are thinking, oh, I'm never going to get where I want to be. You will get there. Just don't be afraid to take the unobvious opportunities that come your way. So I've been just so fortunate to have great opportunities and, importantly, a really fantastic team. I do direct the International Canine Genome Project, and I've published lots of papers, and I have gotten lots of awards, but honestly, they only mean something in the context of the trainees. And to be absolutely clear, every experiment I will show you today has been done by a trainee or a technician in my lab. I am not at the bench scouring and struggling, and I get to do all the fun stuff now. And they are the people who are really need to be credited more than anything today. I have to say, you also have to have time for other things in your life, and I am a nationally competitive and now world competitive Masters powerlifter, silver medal in 2024, and a gold medal this year. I'm going to the World Championships. So South Korea, December 7th, you can look for me. All right, so on to dogs. So dogs were domesticated, and this may surprise you, but only 20 or 30,000 years ago, likely in northern Eurasia, and they are the only carnivore, the only large carnivore to be domesticated. So 20, 30,000 years ago, that is an evolutionary drop, oh, something happened here, an evolutionary just droplet in the bucket. Dogs with jobs, if you think of sled dogs or guardians, we started seeing that about 10,000 years ago. Dog breeding in earnest about 5,000 years ago. Think of things like the Central Asian Shepherd, which were great draft dogs, or the Sluey, various hunting breeds. But if you go to the dog park today, I can guarantee you that almost everything you're seeing, those breeds were developed during Victorian times. Most of the breeds around today were developed within the last 200 or 250 years. So we've taken that drop in the bucket and then we've made it, I don't know, you're physicists, mathematicians, what's the tiniest little thing out of a drop? It's something much, much smaller than that. So things that have happened very, very recently. And additional breeds are being created all the time. So every year, the American Kennel Club adds breeds to their roster. And the explosion of breeds during Victorian times was not driven by human necessity. It was largely driven by aesthetics. Specialists and fanciers said, we want a dog that looks like this. We want something this size, this fur, that fur. And that continues now to this day. And there are now over 350 internationally recognized modern dog breeds. So a couple things to keep in mind during today's lecture. All dogs are members of the same species, Canis lupus familiaris, and as I will say over and over, breeds show amazing levels of diversity. And the example at the top is one of size, where the largest and the smallest of the breeds, what you're looking at here, the Great Dane and the Chihuahua, differ in size by about 40-fold at the skeletal level. And down there we see some nice behavior by that Sheltie as he's herding up the ducks. But within a breed, as shown by the Dalmatians, there's a tremendous amount of homogeneity, both at the level of how they look, but at the level of their genome as well. And we'll take advantage of that as we talk. My lab is in a long-term partnership with dog owners, breeders, veterinarians, lovers. We don't kennel any dogs. We don't breed any dogs. We don't do surgeries on any dogs. We don't advise people how to breed their dogs. We don't do any of that. Ours is truly a collaboration with citizens like all of you who come to us and say, hey, let me tell you about my dog. That's how the conversation usually starts. And that's fantastic. That's how we want to do science. And dogs get the same disorders as humans, and that offers great opportunities for studies of genetic risk. What we learn in dogs is applicable to humans. Over here on the right is oral melanoma in a dog, and on the left is oral melanomas in a human. And I'll show you some other examples where A and B give information to each other. Now, this is one of my favorite slides. I've shown it a million times, I think. It's work by Heidi Parker. And what she's done is she's asked, how do all the modern breeds relate one to another? So this particular slide has 160 breeds. She's now done it with over 300. But just for illustrative purposes, I'll show you this one. So she took 160 dog breeds. She got 10 dogs of each breed, got their DNA, and then she interrogated each one at 170,000 random positions in the genome. And then using all the appropriate computational methods, she drew this neighbor joining dendrogram that organized these breeds into their most likely relationships. And you get 23 what we would call clades, each indicated by a particular color. Now, this is one of the most important things my lab has produced, and the field really relies on it, and we rely on it, which is why we're ever expanding it, because we need it to drive our hypotheses or to develop our hypotheses. So for instance, let's say you're trying to map a gene for epilepsy, and someone comes into your research office and says, well, I have a French bulldog with epilepsy. Well, you can't do much with one dog, right? But you can go and you can say, well, I'm going to collect epilepsy samples from boxers, from bull terriers, from miniature bull terriers, and I'm going to analyze them. And those that have epilepsy likely have it for the exact same reason that French bulldog that walked into your office does. And that's because they share a very recent common ancestor. And you can do the same thing over here with that Scottish Terrier that walks into your office. Not much you can do with one Scottish Terrier, sometimes not much you can do with one breed if they're really an inbred breed. But geez, all these are terrier breeds, and so you can collect DNA from all those. The West Highland White Terrier, and I can't see them, the Norfolk Terrier, the Norwich Terrier, the Wire Fox Terrier, et cetera, et cetera. And those affected ones will likely have the exact same mutation as that initial dog, because again, they have a recent common ancestor. What they have, though, will be different than what you saw over here in the bully breeds. And that's how we solve sort of these complex problems that plague human genetics, where there's just so many genes out there, and people don't come with good labels that tells us, I need to consider you together, and then you together, and then you together, whereas dogs come with great labels in the form of breeds. So today we're going to consider 3 topics: identifying variants associated with morphological traits, variants associated with disease risk and mutation accumulation, and finally we're going to talk about behavior and the genetics of behavior. So morphology first. So when you think about dogs, one of the first things you think about is how big is it? When I say to you, what's your dog like, you'll say either what color its coat is or how big your dog is. And so Joss Plessé in my lab has spent a lot of time identifying the genes for body size in dogs, and here's a primary set of them here. Now, what's so striking about this is there's about 20 different loci. And when I say loci, I'm just talking about a stretch of DNA that has a gene or genes that we know are going to be important in that trait. So there's about 20 loci that control that 40-fold difference in size that make big dogs big and little dogs little. Do the same experiment in humans, which has been done, and you get over 12,000 loci that are the difference between being 5'6" and 6'6". Humans have had a lot more evolutionary time. Dogs have not. So evolution had to hit it fast, hit it right, and didn't have a lot of arrows to throw. And that's exactly what's happened here. So a reoccurring theme when you study dog morphology is that most traits are controlled by small numbers of loci or genes of very large effect, and the opposite is true in humans. Now, the traits that we study, you might ask, why does NIH care? Well, they care because they inform human biology and human disease. So on this bottom panel, I've listed a bunch of morphologic traits and the associated gene that we found. So think about something like the English Mastiff and the Great Dane. They actually weigh about the same. But the English Mastiff is a very big, bulky dog, and the Great Dane is, of course, a much longer, leaner dog. Just even think about their leg length. Well, the difference between being that bulky or lean is due to mutations in ACSL4. That's great. That's what dog breeders have been going after. But when otherwise mutated in humans, it's associated with disorders of lipid biosynthesis. Ear position, we love studying ear position dogs. They can be up, they can be down, they can be the front of the head, they can be in the back of the head, they can be big, they can be small. I could go on for 5 minutes on this. But those genes, MSRB3 and KCNQ5, when otherwise mutated in humans, are associated with various forms of deafness. And this is something that we see over and over. So finding these genes for morphology, which breeders selected on for just purely aesthetic goals, turn out to inform problems in human health and biology. And by the way, these dogs have no particular hearing disabilities. Mutations are in different places. Okay, IGF-1 is probably my favorite gene. It's the first body size gene that we found. It accounts for almost 13% of the variance between being a big dog and being a little dog. So when Joss joined my lab, he wanted to know how is it regulated? Where did it come from? Has it been around for a long time? I mean, wolves are big and it's 20,000 years. So how do we get little in such a short period of time? So he correctly assumed that IGF-1 was part of the story. So let me just start by orienting you to this slide. So there are two variants, one from mom, one from dad. We would say two alleles, and they're called C and T. The C allele is associated with small body size. So if you have two copies of the C allele, you're going to be small. If you have two copies of the T allele, you're going to be big. And if you're a heterozygote, you have one copy of the small allele, one copy of the large allele, then you're going to be intermediate in size. And here's some nice examples when we look at schnauzers and poodles. So when we look at miniature schnauzers and toy poodles, pretty small, 7.5 kilograms for the schnauzers and about 3.5 for the toy poodles. Their genomes are almost entirely red. All the dogs you look at have small. When you look at the mid-size, so for a standard schnauzer, that's about 16 kilograms. For a poodle, not nearly as big, it's about 6 kilograms. But what you start to see is heterozygotes. So you start to see that large allele creeping in. When you look at the big boys, the giant schnauzer and the standard poodle, giant schnauzers are big. They're 36 kilograms. And they are almost all completely homozygotes for that large allele. Poodles are sort of a third, and a third, and that has to do with how they're bred. So poodle breeders, in the poodle world, coat color is everything. That's what really matters. And so they will cross miniature to standard, standard to small, small to miniature, miniature to toy, in order to achieve that perfect coat color that they're looking for. So with that figured out, I should tell you that when I say allele, it's actually a little piece of DNA that's encoded for in the genome. But what it does is it actually binds part of IGF-1 and keeps it from transcribing and doing its job. And so and then that little piece of DNA itself has a mutation in it, just to make things extra complicated. And it's those things together that give you the small or give you the large allele. And so Joss asked, well, where did it come from? Why would you need a small allele if you're a wolf, right? I mean, how does that make any sense? And so we worked with Gregor Larsen at Oxford and Laurent France in Germany to try and tackle this problem. And we analyzed their data of 33 ancient genomes. Actually, many people contributed to that data set. And if we look over here, triangles are wolves, circles are dogs, and red is small, same color coding as before, and yellow is big. So looking over here, we see either yellows, deep yellows for these wolves. We see some oranges, but by and large, those aren't very old wolves. They're like, 0.1 or 16.9. I mean, they're not actually very old wolves. However, there's one wolf that turns out to be really important, and it's this one right here. And this wolf, who's a heterozygote, so he has the small allele, He's 52,000 years old. 52,000 years old. 30,000 years before dogs were even domesticated. Why? Why would nature keep that in her back pocket? Albeit it's at a very low level, but you see it. And so it's a puzzle, and I don't think we really do understand it yet. But we do understand, though, is that nature made good use of it. So if you look here in the Middle East, again, the circles are dogs, and you see lots and lots of these dogs from 1,000-2,000 years or so up to 5,000 years ago, and they're homozygotes for the red. They're little, and in fact, they're little dogs. So somehow nature had that in her back pocket and pulled it out, and then very, very quickly used it. And now suddenly you have small dogs. When we look at other canids, it's an interesting, but maybe it's an expected story. So things sort of north of the equator, like gray wolves in North America, are by and large, they're going to have the large allele. And it's really when we look further south that we start to see these wild canids. So things like the Andean fox or golden jackals, Ethiopian wolves, side-striped jackals that have the small allele. And in fact, if you've ever had a chance, most of these canids in places like South America and Africa are actually pretty small. And that works to their advantage in that environment. Okay, so let's move on a little bit from there and talk about Rubin's work. So we've been thinking about finding a phenotype, finding some kind of a marker, and then figuring out what the underlying genes and mutations are. So when Rubin came to the lab, he said, I want to do it differently. I want to start by finding a variant that I don't know what it does, and then I want to try and figure out what it's associated with. He's a bioinformatician, and he said, traits, that's kind of like biology, ick. He said, but the statistics, the variance, the numbers, I can pull lots and lots of those. So the idea here is to look in regions that are evolutionarily conserved and by and large would be pretty intolerant to a mutation, to the degree that if you had a mutation, almost certainly it would be doing something. So we prioritize variants on constraint metrics. We had high-quality reference genomes. We had at least 3 dogs per breed. We also had mixes and village dogs. And a lot of this came out of the Dog 10K project, but 3 of us had started. And the idea was to sequence thousands of dog genomes and put them out to the community for everybody to use prior to publication, which is something that we've been doing. So then we ranked those candidate variants by breed enrichment. And so I know this just looks like alphabet soup here, but these are some of the genes that came out at the top of our analysis. And I have one squared off here, which is PDGFRA. So this is a gene that it does lots of interesting things, but most importantly, it plays a major role in midline closure in the face. And we only see it in one breed, which is a cattle Burren breed. And part of the standard for that breed is to have this bifid nose, which is really remarkable. This is a good picture of it. These are others, all of which have this bifid nose. We see gargoyles in buildings, in castles, back to the 1500s that have this gargoyle nose, that have this bifid nose structure. So here's some nice mouse data from Phil Soriano's lab, and it shows the presence or absence of mutation in the gene, and you don't get full closure when you have the mutation. All the dogs are heterozygotes. There's no homozygotes. We're assuming that's embryonic lethal. We put out a call for other dogs that were not cattle Burrens to see if we could, find some sporadic ones, and we sure did. Others of them had mutations in different parts of the gene, but not all of them had mutations in this gene, telling us there are other genes out there that remain to be found by continuing this experiment. We looked in the human literature and we found a pedigree who had a mutation in this gene. So individuals who are colored in black have cleft lip and palate, and they all carry the mutation, which is the star indicates. So everyone who has it has the mutation, but there are some people with mutation who don't have cleft lip and palate. So that means by following up in this canine system, we have this great way to try and understand genetic penetrance. We all know some things do their thing in certain genetic backgrounds and not in others. Not every woman who has a BRCA1 mutation will get breast cancer, right? And we need to understand what are the other genes that contribute. Genetic background, the role of genetic background in expression continues to be a really, really perplexing and interesting problem. So we have an opportunity to do this here by looking at this phenotype, this evolution that humans did, again, because this is something they wanted to see aesthetically. Now, when I give this talk, people often come up to me and they go, not a problem for me. I have a mutt. It doesn't matter. I have hybrid vigor working on my side. Well, it depends on the health of the parents, because purebred dogs can pass on purebred problems to their mixed breed puppies. And there's lots of examples in the literature relating to a variety of diseases: exercise-induced collapse, congenital ichthyosis, diabetes, various veterinary ophthalmology problems that come up in mixed breed dogs. So we decided to look into this just a little bit because we were kind of curious about this. And the bottom line is mixed breed dogs or mutts are only as healthy as what goes into them. So this is a collie, and about 69% of collies have a gene that makes them sensitive to various drugs. The gene is called MDR1, and for instance, it affects the ability of these dogs to handle heartworm medicine. This is Grace. She's owned by a friend of mine. That dog is 12% collie, so it probably has a pretty low risk of drug sensitivities. But this is also owned by a colleague of mine, Sophie. She's 73% collie, so she has a very high risk of drug sensitivity, something her owners may not have even been previously aware of. American Staffordshire Terriers have about 26% have hip dysplasia. French Bulldogs, that number is 33%. This is Hattie. Hattie is 25% American Staffordshire Terrier and 12.5% French Bulldog. And so she actually turns out to have a pretty moderate risk of hip dysplasia. Again, something her owners may not have been aware of. And people ask, how big a problem is this? Because so many of us do go to shelters in order to get our dogs. And it actually is a big problem. So in 2011, a paper was published describing 152 disease gene causing variants. And most of this data, and it was a literature summation, had come from studies of purebred dogs. But then in 2018, Donner et al. Published a paper looking at 100,000 dogs, and 80 variants, disease-causing variants, were seen in his compilation of purebred and mixed breed dogs, but there were 34 that were not seen in purebred dogs that were seen in mixed breed dogs. So breeders had cleaned them out of purebred dogs, but they were still hanging around in the mixed breed dogs. So genetic testing allows breeders to eliminate disease variants in purebreds, but crossbreeding has allowed the variants to remain. That's something to think about. All right, so I just, before I switch on to cancer, I did want to say a word about Labradoodles. So they're a really interesting breed to me, and when Dr. Basil Ali came to my lab, we decided to look at the Australian Labradoodle. So this was an Australian poodle cross from 1989, and the idea was to make a service dog that was allergy and asthma friendly, low shedding. To be a purebred Australian Labradoodle, which is an actual breed, you have to descend from 5 or more generations of Australian Labradoodle crosses. Now in the US, if you have a Labradoodle, it's probably a first-generation dog. This is different. This is like multiple generations, and this is a true breed. So we looked at first-generation crosses and dogs from generations 4 through 8. Let me say at the onset, these are actually pretty healthy dogs. They live to be about 15. They do have some, progressive retinal atrophy, some hip and elbow dysplasia, some patellar luxation, but actually they're a pretty healthy breed. But when we looked at them, here's what we found. So when you compare the poodle to the Labrador, and you start at the top of chromosome 1 and you look all the way to the bottom of chromosome 38, dogs have 38 chromosomes, plus the X, and what we're looking at here is allele frequencies. You see lots and lots of red. And that makes sense because they're two very different breeds of dog. So allele frequencies should be very different at lots of SNPs or single nucleotide polymorphisms or markers as you look across the genome. When you compare the Labrador to the Labradoodle, you see half as many. It was about 6,000 up here. It's about 3,000 over here. And that makes sense because Labradoodles have, obviously, Labrador Retriever in them. When you look at the Poodle versus the Labradoodle, you see almost nothing. You see only 5 that are significant. So that's telling you that in a strong desire to breed for a particular kind of coat, hypoallergenic, not shed, the Poodle genome has completely dwarfed out the Labrador Retriever genome. And your Labradoodle, if you have an Australian Labradoodle, is actually a lot more Poodle than it is Labrador Retriever. Great dogs. I'm a big fan of them, actually. But we don't always have what it is we think we have. So I'll move on. There's lots of different things that we've looked at: body height, skull shape, leg length, coat color. Whatever you're interested in, please come and talk to me, and I'm happy to share anything we have with you. Let's talk just briefly about cancer. So many breeds have an elevated risk for specific cancer. It's the #1 killer of dogs. About 45% of those over 10 will die of cancer. This is not something we want to have happen, but again, strong selection for traits is allowing these deleterious things to carry along in the genome. We see a lot of osteosarcoma in the long limb breeds. We see bladder cancer in Scottish, West Highland White Terriers, and in Shetland Sheepdogs. We see malignant histiocytosis, histiocytic sarcoma, as I told you, in the Bernese Mountain Dog. The story is the same in the flat-coated retriever. About 20% of those will get it, and those that get it will all go on to die of the disease. And gastric cancer, it's not a common disease, but it's really lethal, and we see it in a number of the Belgian breeds as well as in the Chow Chow. So my lab has been working on invasive urinary cancer in the dogs. It's not big in terms of percent, but there's about 50,000 new affecteds each year. About half of those metastasize to distant organs, and those dogs have an average survival of about 1 year. And as you may know, this is not a good disease to get if you're a human as well. Common treatments are pretty much as they have been for dogs for the last 20 years, no real advances. But what is fascinating is this. If you are a Scottish Terrier, your odds of getting invasive bladder cancer are 21-fold higher than the average mixed breed walking down the street. 21-fold higher. You just don't see that in human genetics. So this is a great opportunity to study a disease that we haven't made tremendous progress in humans. And it has to do with the way breeders have been crossing dogs for, again, specific aesthetic traits. American Eskimo dogs, it's 6-fold higher in the Shetland Sheepdog, which is what we're going to talk about. It's about 6-fold higher. So we did a genome-wide association study in the Shetland Sheepdogs and in related breeds. And again, we're looking from the top of chromosome 1, the bottom of chromosome 38 in the X, with every dot representing a marker or a place in the genome that we really focused in on. We found 3 adjacent, we call them SNPs, single nucleotide polymorphisms. They're just places of variation in the genome on chromosome 13 that gave us a signal above the background. With a p-value of 10 to the -7th, even after correction for population structure, the result remains significant after 10,000 permutations. But then we said to ourselves, well, we had that wheel. What about that wheel we showed you at the beginning? Take advantage of what we know about breed structure. So we added in data from affected collies, from affected Australian Shepherds, and from affected Border Collies. And now the signal-to-noise ratio is much cleaner, and the p-value goes from 10 to the -7th to 10 to the -10. So just knowing how breeds relate one to another gives us so much power to understand what dog breeders have done and how we can help humans. Now, this isn't a slide I put up for you to read everything. I just wanted to make one quick point here. So Nepal-1 is the critical mutation that we found here. It's overexpressed in bladder cancer cell lines. Risk is increased in the homozygous state. But we also found other genes that increase risk or decrease risk, as it turns out, on chromosome 28, 4, 9, 21, and I think one other. So this incredibly messy graph is just telling you that it's combinations of the different variants in these genes that can either increase your risk, can decrease your risk, or don't affect your risk at all. And that's why cancer is a really hard problem. That's why you have families where some people get it, some people don't. There's a trend, but it doesn't really, fill out statistically well. There's a population that gets it more than other populations, but you can't find the gene. This is why in a microcosm. We can explain 70% of the risk in this breed with these 5 or 6 genes, 70% of the risk, right? So that's fantastic. And it's a really much harder problem to solve when we look at humans. But we're obviously continuing on this route. And everything now is about the Scottish Terriers. The problem is, there's things in the Scottish Terriers that are obviously fixed that you just have because you're a Scottish Terrier. And that's confusing us. I mean, it's a beautiful dog. It's a really, really beautiful breed. But we have to solve that problem. All right, and so in the last 15 minutes, I decided I wanted to talk about behavior. And we are doing work in my lab on anomalous behaviors, but I did want to talk about sort of what a lot of people would call stereotypic behaviors. So when I say breed behavior, a bunch of pictures come to mind. We think of herding dogs that herd, sled dogs that pull, pointers point, draft dogs pull, retrievers retrieve, and sighthounds, of course, play the role in hunting. So these images come to our mind. Now, let me just say that because you are a given breed doesn't guarantee you're going to have a certain behavior. So my border collie, Tess, is the world's worst herding dog. I will readily admit that, for sure. Even though I watched both her parents herd out on a farm, and they were both fantastic. She is not. Great dog, not a good herder. So it's not, you have to take that, keep that in mind, because that's an important caveat to all this. When we talk about instinctive dog behaviors, we're talking about breeds that were developed because we needed them to meet some of our needs. We needed those behaviors in order to help with our own survival. The word instinct is used by us to describe behaviors that occur early in life upon the occurrence of a specific triggering stimuli and that occur naturally without any prior training. So in this case, the dog is looking at a herd of sheep, and that's the stimuli that's getting her to crouch and show eye and all those things. I had a video, but I wasn't sure videos were going to work here, of three Shelties, and the very first time they saw sheep. They were 10 weeks old. It was the very first time. And it was spellbinding because they were herding the sheep. They were working as a team and driving them here and driving them there, and they had never, ever seen a sheep, not even one walking by. It was just remarkable. And you can't look at something like that and say there's got to be some genetics going on. There's a lot of other things going on, but it's really remarkable when you have a chance to see something like that. So Emily Dutro was an incredibly talented postdoc who came to my lab. And what she did was to build a high-dimensional representation of canine genetic data with FATE, and the goal was to identify genes that were associated with lineages of behaviors. So FATE was originally developed for looking at single-cell data, and this is actually the first time it had been applied to a big population of genetic data. But when she did that, she got exactly what she'd hoped. She got lineages. So all the dogs on this line of the star come up through to the retriever lineage. This is the terrier lineage, pointer spaniel lineage, Centhound. This is African and Mideastern, and so on and so forth. And right here, this big Gamish in the middle are, less, I would say, less. I don't mean distinguished in the sense of being distinguished, but I mean they're not as diversified. They're not as singularly diversified. They're a more homogeneous group of individuals. So we pulled out the herder lineage. So the herder lineage has lots of different kinds of dogs who do things related to herding. So it has the actual herders, it has healers, it has drovers, it has boundary dogs, and it has guardian dogs. So all dogs that have some kind of a role in herders. And then Emily looked specifically at the herding dogs, so things like border collies and Shelties and those guys, Shetland sheepdogs. So we're going to look at them in a little bit more detail here. So here's what she found. I should say one other thing. Let me go back. So the reason fate was so important to this paper is because unlike other things, fate allows you to have unlimited neighbors. So like a phylogenetic tree does not allow you to do that. Fate allows all possible data structures. They can be bifurcating, they can be clusters, they can be linears. And so we could, in a much more real sense, understand the relationship of these trajectories of breeds and how they moved along and became more and more specific and more diversified, with the breeds at the end of array being the ones that are most differentiated. And then obviously here that's going to be the actual herding dogs. And so we looked at that in more detail. So Emily did an enrichment analysis, and it pointed her towards pathways important in axon guidance, those things that tell axons where to grow, because you have to be in the right place, whether it's whatever part of the brain or, wherever it is you're supposed to be, you have to be there in order to do the job that you would do. My husband's a neurobiologist. He's going to hate that when he sees that. That's how I described axons. But you really do. And so this is, again, one of those genome-wide association looks. And the genes in green are the ones that she's paying attention to. And when we look at them in their pathways, we can see that they identify really 3 classes of axon guidance genes. Those important in outgrowth, those important in attraction, and those important in repulsion. So that was really exciting because even as we start to break down the genes, they're highlighting much more specific pathways. So this is Ephrin 3, and I've just stretched out the chromosomes, and you can see there's sort of a fountain of dots here, each one being a different marker, and right around it, it's absolutely flat. This is Ephrin 5, and you see the same sort of fountain here, and this is Slit 2 with the same sort of fountain. So many of these genes are actually part of Ephrin signaling pathways. Well, what does that mean? How could Ephrin contribute to herding? Well, for instance, EPHA5 and its ligand have been implicated in anxiety and maternal pup gathering in mice. So collect and contain and keep them all together like the mom does. So we speculate, without a shred of data, that herding drive could involve augmentation of those same ancestry, anxiety-associated pathways that drive maternal protective behaviors. I don't know. We're going to have to test those hypotheses. Shortly after this paper came out, actually this year, I was thrilled to be able to work with Jae-Min Kim's lab. Jae-Min had been a postdoc in my lab. He's now tenured faculty, has his own lab. But we were able to show that another Ephrin, EPHA1, mattered as well. And what he did is he compared border collies that had bred for show versus those that were bred for work. And they look very, very different, and they act very, very different. And he showed that variants in Ephrin 1 distinguished them. And so EPHA1 is linked to locomotor activity and spatial memory. And he saw this really clear evidence of segregation within these 2 lineages. So an evolving story that Dana Drager in my lab is continuing to work on. Okay, so in this last vignette, I want to revisit something that we did several years ago. And this was also led by Jae-Min when he was in my lab. And he was interested in looking at groups of breeds. And so he did a lot of sequencing and sequenced lots of sport hunting breeds and terrier breeds. And they represent the ends of an athletic continuum. So sport hunting breeds, they're bred to assist hunters by retrieving, flushing, pointing. Their job requires very prolonged periods of physical activity, whereas terriers are employed to rid land or buildings of vermin. That's actually even what they do still today. And they need very rapid, short bursts of activity. Run, get it, bring it back. Run, get it, and bring it back. And they're actually barn hunt competitions where this is exactly what they're doing. They're looking for vermin, and you get points and ribbons and stuff, if you find them. So, But what they do is very different, but both kinds of dogs are very athletic. So Jae-min looked for signatures of selection to see what mattered to be successful in these two groups and what differed between them. And we found genes that mattered were either neurological, so things that were important in humans in learning. Other genes that when mutated in humans were associated with mental disabilities. Lots of muscle mattered. So things that were important in muscle contraction, muscle development, fatigue-enhanced muscle pain. So whether or not you perceive that buildup of lactic acid. If you don't, it doesn't really matter that it's there. Genes that when mutated are associated with muscular dystrophy in humans and things that affect muscle mass. And not surprisingly, cardiovascular was a big one. Heart rate, hypertension, vascular smooth muscle contraction. And it was great to find these three categories because Joyner and Coyle had predicted in 2008 that elite athletic performance was largely determined by the integration roles of muscle and cardiovascular and neurologic functions. So we were right where we expected to be. So let me talk about myostatin and TRMP3. So we had shown a couple of years earlier that if you knock out myostatin in whippets, you get a much more heavily muscled dog. So if you knock out myostatin, we did not knock it out. This is a naturally occurring mutation. We don't do that kind of thing to dogs, but it is a naturally occurring mutation. And they also had the relevant mutation in TMPR3. Those whippets ran 27% faster than the same whippet who didn't have those mutations, who might have been from the same pedigree, raised in the same place, trained in the same way. So just those two, knock out myostatin to get more muscle, and TRMP3, and you can increase racing speed by 27%. That's remarkable. Now, these myostatin knockouts do occur naturally. This is a heterozygote, and you can see this is a beautiful dog. I mean, this looks like a racehorse with all that well-defined musculature. The dog down here is a homozygote, and it's just too muscle-bound. It's never going to be really successful on the racetrack, right? And I will say a massive amount of data went into these experiments to get, racing data and racing times and different tracks and all that sort of. I mean, it was a huge experiment to get all this data. I also wanted to say that when we first published the myostatin result, it was really close to an Olympic year. And I got inundated by the media saying, well, we should test all the sprinters at the Olympics, and those that have a myostatin mutation, they should race separately than everybody else, right? Would you go on record as saying that? No, I'm not going to go on record as saying that. Of course not. There's lots and lots of these, I would call them performance-enhancing polymorphisms. I call them PEPs. That's a word I, made up. But there are a lot of them. And this shows how it's a conglomeration of things that come together in order to get certain behaviors. So we also thought a lot about ROBO1, which is another really interesting gene. So ROBO1 is associated with success in the obstacle course in dogs. So obstacle course is called agility in the dog world. So you can think of it as American Ninja Warrior. You jump, you go up, you go down a slide, you go through something. A lot of start, stop, really fast motions that you have to do. So we took a huge number of dogs from 92 different breeds, and we pulled out the top most successful 20% and the bottom least successful 20%, threw out all the squishy data in the middle, and then looked at this large set of genes. And ROBO1 was significantly associated with breed-specific agility performance. And again, that was even correcting after we corrected for morphologic traits, like if you're big, if you're a breed that's big, or you're a breed that has long legs or short legs, even after correcting for all of that. So this was just really such a cool result. I was so excited about this and still am. ROBO2 is a gene that plays a role in neuronal migration, development, and again, axon guidance and growth. In humans, it's associated with developmental dyslexia. So it's a learning disability associated with translating visual cues. And so that makes sense when you think about what you have to do in agility or an American Ninja Warrior, right? You have to be able to turn things around in your head or determine how far away they are from you. And you have to remember that. I've seen something like that in that size. It's probably going to require me to do this or rotate this way rather than rotate that way. In dogs, it affects their ability to identify and learn environmental information, such as what you see in an obstacle course. So we have a lot more behavior stuff that we're doing. It's all just so incredibly fun. But it's shown us that it is very complex, and it's going to be this interplay of morphology and neurobiology and cardiovascular function. All these things have to come together in order to have a dog execute a behavior successfully. And so just like the cancer story, this is going to be hard, but it's going to be easier than doing the same thing in humans, or at least we believe that. All right, so I'm going to stop there because I've given you a whole smorgasbord of things that my lab is currently doing. I hope I've convinced you that early dogs, we are responsible. We did the experiment. In fact, I always argue that the development of dogs and domestication of dogs is the most successful experiment that humankind has ever done, because we needed them to survive, and we did it right. We did it successfully. Most modern breeds were developed during Victorian times in just the tiniest drop in the bucket. I mean, think about the dog park, and you're looking at 200 years. It's like nothing. So all the genetics that was embedded in those wolves' genomes, and I showed you the example with IGF-1 small allele. I don't know why all of it's there, but I know it's truly, truly remarkable. And that, for the next generation of dog geneticists, has to be one of the most provocative and interesting questions. Evolutionary constraint and comparative genomics certainly deepen our understanding of canine traits and disease. And you know, I'm partly intrigued by the bifid nose thing. It does exist in one other breed, a Spanish breed, but we've never been able to access samples from that breed. So only 2 breeds have it, and then you have a few sporadic dogs that have it. And yet, for some reason, it's been under selection here. In humans, the same thing is really deleterious. And that just crystallizes for me why you study things under selection in dog breeds, why that informs human health and human biology. Dog breeds and knowledge about breed relationships serve an important role in finding trait-associated genes. Small numbers of genes of large effect control many complex traits in dogs, and that is very often our selling point in writing grants and proposals. Anyone who wants to access our data, especially all of our sequencing data, which we've made publicly available, can scan this for data access or look here. And I forgot to put our website up here, but send me an email and I'll certainly give you our website and our Facebook page, which is where we put all of our really current updates about what's going on. So I'm going to stop. I tried to make a slide acknowledging all my collaborators. And after I was on the 3rd page of that, because I've had so many generous collaborators, I just decided not to, because I didn't want to end up omitting important people. And I just wanted to, again, focus on the people in my lab. These are the people who are in my lab, or actually, Gabby just graduated. She's been doing the work in Chernobyl, where we have a large cohort of dogs that we've been following. And this is Ruben. I was just telling you about Ruben. That's him there. And some of these people are doing projects I didn't even talk about, like looking at ancient dogs in Greenland, which Tatiana just had a paper in Science about. And these are people whose work was included in my talk today, but have moved on to do other things, many of them to run their own labs. And I always have to thank the dogs, and I have to thank their owners, who are just so generous with their time and resources and data that they give us and share with us. So right on time, I'll stop there.